Today's video lecture topic is going to be on the importance of RNA and the process of protein synthesis. I'm going to warn you now, this is one of the hardest topics that we cover all year, if not the most difficult topic. So pay close attention, pause when you need to, rewatch as many times as you need to make sure that you understand this. Make sure you're filling in your notes organizer as you watch the video. Make sure you answer all questions so that you can use your notes on the notes check. Okay, so the first thing I want to discuss with you is this huge idea in biology. That's why we call it the central dogma. It's just this sort of broad concept that all the rest of biology is based off of. So the central dogma of biology says that DNA, which we know is the blueprint of life, the carrier of genetic information, is read by RNA, which then travels to the ribosome to make proteins. So it's just this big idea that the instructions are carried on DNA, those instructions are read by RNA, where then they're brought to the ribosome and turned into proteins. Okay, so that's the answer to number one, and you can just write what's down here at the bottom. So we start with DNA, it's read by RNA, and then it's used to make proteins. That's the central dogma of biology. This huge idea is what the rest of biology is all based on. This is why living things exist. So we've talked a little bit about this in class, but we know there are major differences between DNA and RNA. So RNA, just like DNA, is made up of those subunits called nucleotides, but there are four major differences between these two molecules. So obviously there's a letter difference, DNA and RNA. The reason they have different letters is because they have different sugars. So RNA has the 5-carbon sugar called ribose. DNA has the 5-carbon sugar called deoxyribose. They're essentially the same, but deoxyribose has been deoxified. It's without an oxygen, or it's lacking one oxygen. Okay, so we know the bases in DNA are A, C, T, and G. RNA has A, C, and G, but instead of thymine, it has a base called uracil. Okay, so it's a pyrimidine, just like thymine is. It takes the place of thymine. Still pairs with adenine. RNA is single-stranded instead of double-stranded, so it is literally smaller than DNA, which leads to our next difference, that RNA is able to travel in and out of the nucleus. It's literally physically small enough to travel in and out of the nuclear pores. There are three types of RNA. One is messenger RNA, which is the messenger. It carries the message from the DNA that's in the nucleus to the ribosome so that it can be turned into proteins. Then there's ribosomal RNA, and this is the one we sort of forget about. It's just RNA that makes up the structure of a ribosome. A ribosome is literally jumbled up rRNA, ribosomal RNA, and some proteins. That's what a little ribosome is. It's not a fancy organelle. And then the last type of RNA is called transfer RNA, and its job is to transfer the amino acids to the ribosome that are going to form the protein. Okay, so you have mRNA, messenger RNA, carries the message, ribosomal RNA makes up the ribosome, and then transfer RNA transfers the correct amino acid to the ribosome so that it can build a protein. Okay, we're going to talk about two processes today which make up the overall process of protein synthesis. So transcription is the first process, translation is the second process. They sound similar, they're both complicated. So you've got to take the time to make sure you know these two processes. Okay, so first up we have transcription. So think of the genetic code on DNA as your recipe. That's your grandmother's favorite recipe. We have to get your grandmother's recipe to your kitchen, right? So there has to be some copying down of that recipe. That's exactly what's happening in transcription. We have to copy the message that's on DNA onto a molecule of messenger RNA so that it can leave the nucleus and go to the ribosome. Okay, so big picture, the process of transcription is the making of RNA from DNA. This is obviously taking place in the nucleus because we're dealing with DNA anytime DNA is found, it's found in the nucleus. And here's how it happens. RNA polymerase binds to DNA and begins building a complementary strand of mRNA. So we know what DNA polymerase did. It put on new molecules of DNA, right, or new nucleotides to build a molecule of DNA. RNA polymerase is going to use the template strand of DNA in order to build a molecule of RNA. So now adenine is going to pair with uracil, but if a T is present, it's still going to pair with adenine. Cytosine and guanine go together. So why is this happening? What is the big idea? We have to get the message from the DNA in the nucleus to the ribosome where the protein is going to be built. Remember, messenger RNA is smaller. It can leave the nucleus. So here's how it happens. A little gene opens up. Okay, so a piece of DNA opens up, and that RNA polymerase enzyme starts constructing that complementary strand of DNA. So this complementary message here is going to travel through the nuclear pores and head to the ribosome. So a couple things you need to know. DNA and RNA are read 3 bases at a time. That 3-base sequence is called a codon. A codon signals for a very specific amino acid. There are 64 possible codons, 3-letter combinations. Remember, you've got 4 letters. So of those 4 letters, 3-base sequences, there are 64 possible combinations, but there are only 20 different amino acids. Okay, so there are several codons that code for the same amino acids. Remember, the building blocks of proteins are amino acids. The building blocks of proteins are amino acids. You learned that. Now we're going to learn about these amino acids. How do they come together to form proteins? So amino acids are the subunits that come together to build a protein. So let's do this together. We're going to practice transcription. If this is our DNA sequence, AGC, there's our first codon, TCC, GAT, blah, 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 can you figure out what the RNA complement would be during the process of transcription? What would be the sequence of the RNA molecule that the RNA polymerase would build? Let's see if you can get it. Take a second to see if you can figure it out. Okay, so our complementary sequence would be UCG. AGG, CUA, blah, blah, blah. Do you get why? Adenine, remember in DNA it pairs with thymine, but thymine doesn't exist in RNA. So adenine is now going to pair with uracil. Guanine still pairs with cytosine. Cytosine still pairs with guanine. Thymine still pairs with adenine, right? This is your DNA molecule. This is your RNA molecule. We still have adenine. Okay, try the next one now that you have an example here. Do you get why this is the answer? Do you get why we have uracils on our RNA sequence instead of thymines? Okay, so we are about on number 9, 10 on your notes organizer, so please make sure you're keeping up. Okay, so here is our RNA sequence. This would be after transcription. That is going to get separated into codons. And each of those codons is going to signal for a very specific amino acid. Okay, so if those were our codons, so our first codon is UCG, you have to use this fancy little codon chart to figure out what that signals for. So this chart is really cool. You just start in the middle and then you work your way out. So our first codon, if you remember back over here, was UCG. So start in the middle, UCG. That codes for serine, the amino acid. That is the amino acid that is signaled by that codon. Okay, see if you can do the next one, AGG. Start in the middle, AGG, that's arginine. Okay, last one, CUA. Start in the middle, CUA, that leads to leucine. Okay, you've got to be able to know how to do this. Start from an RNA or start from DNA is really figure out the RNA complement after transcription, and then what would be the amino acids signaled from those codons. So now that we've transcribed the message, we've copied the message down onto a molecule of RNA so that it can leave the nucleus and go to the ribosome. We have to translate that message. So when you get your grandmother's recipe, you have to start getting the ingredients and putting it all together in your kitchen, right? That's exactly what happens during the process of translation. The kitchen is the ribosome. This is where the proteins are going to be built. This is where the recipe is going to be followed. So the big picture of translation is taking that message on that messenger RNA molecule and turning it into proteins. So it's the making of proteins using the instructions from the mRNA message. In other words, translating what you have just written, translating what you have just transcribed. So we have transcription in the nucleus, and now we're going on to translation, which takes place at the ribosomes. Ribosomes build proteins. You know that, you memorized it. So this is where this process is taking place. Now look, this is complicated. It's not easy. This is what it looks like down here. We're going to break it down into the individual steps. Okay, so transcription has just happened. So DNA message was copied onto a molecule of messenger RNA. That messenger RNA is going to leave the nucleus and enter the cytoplasm. It's then going to make its way to the ribosome, right? This is where translation happens because this is where proteins are built. And the tRNA molecules, remember tRNA stands for transfer RNA, the transfer RNA molecules are going to transfer or bring the correct amino acid. Now, how do we know they're bringing the correct amino acid? Remember, the mRNA had a 3-base codon, which signaled for an amino acid. The tRNA molecule actually has a complementary anticodon. I told you it's complicated. So the anticodon and the codon pair up, and that ensures that the correct amino acid is being brought to our growing protein chain. Okay, after the transfer RNA brings the amino acids are going to join together with peptide bonds. Okay, so the peptide bond is going to form, the next amino acid is going to be brought in, the peptide bond is going to form, the next amino acid is going to be brought in, the peptide bond is going to form. That's going to keep happening until the ribosome reaches what's called a stop codon. And a stop codon does exactly what it sounds like. It stops the process. It signals that the protein has been built and it needs to be released to go wherever it needs to go into the cell. So the completed protein, also called a polypeptide chain, okay, poly meaning many, has many peptide bonds holding those amino acids together, is then going to be released into the cell wherever it needs to go. So you're going to want to probably pause on these last couple of slides and get all that information down. All right, I've got a joke for you. You know my kind of jokes, super cheesy. Okay, the teacher says, what are proteins made up of? And the student raises his hand and he says, mean old acids. Why is that a super cheesy, super funny joke? Write that down on a little scratch sheet of paper and turn it in so that you can get a bonus point on your notes check. Okay, so here is the process of translation. You're going to want to pause on this and label your picture here. So on your picture, you have the ribosome. This is where this process is taking place. You have the messenger RNA molecule, which is being read 3 bases at a time. Those bases are signaling for these guys over here, the amino acids. Those amino acids are going to be brought by the transfer RNA molecule that has the complementary anti-codon. Okay, now what happens if something goes wrong? So what happens if the, messenger RNA doesn't decode the message correctly, or what if the wrong amino acid comes in? Those are called mutations. A mutation is just any change in the DNA sequence that is going to affect the building of a protein. So it's going to affect the organism in some way. Gene mutations result from changes in a single gene. So a gene, remember, is what carries the recipe for a single protein. So the gene opens up, a little section of DNA opens up, that codes for a specific protein. If a letter within that gene is altered in some way, changed in some way, that's called a gene mutation, a very specific mutation. Chromosomal mutations are what we talked about, like with Down syndrome, where you have an entire chromosome that is being affected. So maybe part of the chromosome has been removed, or you have an extra chromosome, or you have a missing chromosome. We're going to really be focusing on those gene mutations here in these next couple of units. We talked about this when we discussed mitosis and meiosis. You know that somatic cell mutations are going to affect the individual organism in that generation, but that they are not going to be passed on, because somatic cells are body cells. It's mutations that occur in those gametes and those sex cells that are going to be passed on to the offspring if that egg or that sperm happens to be the one that's fertilized. And then it's going to be present in every cell of the offspring. Okay, let's get into some types of gene mutations, so affecting single letters. A point mutation is where you literally have one single letter, one single nitrogen base that has been altered. So one type of point mutation is called a substitution. This would be like instead of a G, you accidentally have an A that is placed on a molecule of DNA or on a molecule of RNA. It's going to affect one amino acid and therefore affect one protein. You also have more detrimental mutations called frame shift mutations. This is when you have an insertion or a deletion of a nitrogen base, which then alters the entire reading frame after that. Think about it. If you put one extra letter in, or if you take one extra letter out, everything is going to shift, which means every codon is going to change, which means every amino acid sequence is going to be wrong from that point on. Insertions and deletions cause frame shifts because they shift the reading frame of the genetic message. So the ribosome is reading 3 bases at a time. If you stick an extra base in or you move an extra base, that is going to alter every amino acid that is being sequenced or being signaled at the ribosome. Okay, on your paper you have a sentence that says the big fat cat ate the wet rat. Very similar to this sentence right here. You can see how that reading shift is changed if you have something like a deletion. So I want you to give this a try, but don't stress out if you don't really get it, because we will go over that together in class, but I do want you to try it. Okay, so use this, the big fat cat ate the wet rat, and see if you can copy down a substitution, an insertion, and a deletion. And then test your knowledge with that very last question by putting the items in the correct boxes. But again, don't stress about that one if you're not sure where to put something. We're going to go over that in class as well. I hope you're having a great day. Thanks.